Hi, today we're going to talk about enzymes and energy flow in cells. So we're going to investigate our enzymes, and that's going to be our foundational knowledge. So we're not doing a comprehensive deep dive, we're just covering the basics. We'll preview our photosynthesis and our cellular respiration just a little bit to give you an idea of what's coming. So some things to expect today. We're going to want to make sure we're intentional about our notes because there will be a note check. We'll do a phenomena study, so that's going to be checked as well. You'll have a CER, and we're going to change things up just a little bit with that, where you'll submit your CER through Canva, and we'll do a reflection form, but that won't be graded, but I do want your input, so take it seriously. And as always, this content can and might appear on your upcoming unit test. All right, so we're going to start right off the bat with our banana phenomena study. Each table group is going to have one banana slice, and we're going to revisit or echo this phenomena study later today. So make sure you leave room in your notebook for this. We'll set a timer. So you'll start with your initial I notice, I wonder about your specimen, and then in 15 minutes, we'll do a second I notice, I wonder about the same specimen. So here's how I set up my banana phenomena study so that it's all on one page in my notebook. I have my diagram drawing of the banana slice and a start and an after because I know I'm going to be timing this. So start with your I notice, I wonder for your initial specimen. So remember your I notice, those are your statements, what you're observing, and your I wonder needs to be a question you have about the specimen. So your banana slice probably looks like this, and unwrap it. Make sure that there's no banana peel on it because that can be something that you're also going to include in your I notice or I wonder statement. So everyone should have their I notice and I wonder done, and we'll start that timer, and in 15 minutes we'll revisit this. So here's what my initial banana phenomena study looked like. I noticed that the banana is white and has dryish sides with a wet feeling cut. So where the slice happened, it feels different than on the sides of the banana where it was peeled. My I wonder question is, why does it turn brown over time? Because I know what to expect. I've interacted with a banana before. So your notebook should have this. It should be complete sentences or close to it. All right, so on a new notebook page, you're going to want to title it Enzymes, or what are enzymes? Well, enzymes are just proteins, and these speed up chemical reactions by lowering the activation energy necessary to complete the chemical reaction. So sometimes they're going to be called catalysts for biological systems, and something that you're going to want to make sure you include in your notes on this first page, enzymes are specific to their substrate. And we'll cover substrates more, but a substrate is just the reactant that fits into an enzyme's active site. And the substrate is what gets changed during the chemical reaction. So your enzyme is going to be the lock to the substrate being the key. It fits perfectly into that specific lock, but the lock doesn't change. So this enzyme's active site would be where the key fits into. So that's why we use that lock and key metaphor a lot with enzymes. So your enzyme, that's the lock, the substrate is the key, and the active site is where the key fits into the lock. So enzymes are specific to one kind of reaction, but they don't get used up. So it can be done over and over and over again. I mentioned chemical reactions. Well, enzymes are the molecules that affect the rate of chemical reactions. If we didn't have enzymes, one, we would probably die, but enzymes also make it more efficient. We don't need as much energy or as many molecules or atoms to complete a process because of enzymes. So the enzymes are the molecules that change the rate, how long it takes for that chemical reaction to take place. And the chemical reactions, these always need a certain amount of energy to start. That starting energy is called the activation energy. So you need something to get it going. So how do enzymes work? And we kind of already talked about this. Enzymes are going to work, and we'll continue this lock and key metaphor, we'll extend it. So if the enzyme is the lock and the substrate is the key, you have the product has to be released. So you have to unlock your enzyme for a product to happen, for that reaction to take place. So the lock doesn't get destroyed in this. It can be reused after this has happened. So your substrate, that key binds to an active site. You put the key inside the lock at that specific spot. And the enzyme uses a substrate complex to form different things. That's the product. That's what you're unlocking with it. So that's why we love to use this lock and key metaphor in science for enzymes. So enzymes are always working, but they need certain things so that they can function. So what do they need, or what can impact how they function? Well, temperature can change a lot of things. So temperature can, if it's too hot, it can change your enzymes. It can either make them go too fast or not work at all. If it goes too cold, your enzymes, again, they might not work or it might slow them down. So temperature has to be regulated. The pH, this is going to also be an important factor. In your stomach, you have a lot of enzymes at work. Well, if it gets too acidic or too basic, you're not going to have the enzyme keep their shape. And if they don't have the shape, well, that substrate, that key isn't going to work in the lock, the enzyme. So you need the temperature and the pH to both be exactly where they are supposed to be. There are other things that can inhibit or just slow things down besides temperature or pH. You can also have competitive, and that's where the active site gets blocked. And sometimes that's necessary. Other times it's going to be a hindrance and it's going to harm the process. So remember, the active site, if we're extending that lock and key metaphor, is where the lock fits into, where the key fits into the lock. So if you block the keyhole, you can't get the substrate to line up and interact with the enzyme. You can also have non-competitive inhibitors, and they change the enzyme's shape. Well, if the shape is wrong, if you change the shape of your lock, your key isn't going to work. Even if you have the correct substrate, that's not going to fit in the right way with your enzyme. So there's lots of things that can impact your enzymes and how well they function. So what might your notes look like at this point? Well, we should at this point have a basic definition of what an enzyme is. So for my notes, I have enzymes are molecules that affect the rate of chemical reactions. Pretty straightforward. We already saw this. We've heard this. Chemical reactions don't just happen. The reactants must have a certain level of energy for the reaction to happen, and that's going to be called your activation energy. That was mentioned. So we want to make sure that as we go through these notes, we are writing things down. You know I love to include diagrams and pictures in my notes. So I have this not as just a lock and key metaphor, but I also like to think of this similar to riding a roller coaster for that starting process for the enzymes to start working. Well, I know I want to have something happen, so I get on the roller coaster. To get started, though, I still need energy. So that's going to be where you're going up one side of the hill. You have to go all the way to the top. You finally have enough activation energy. And then once you have enough activation energy, the reaction can occur. And you're going to go down that roller coaster hill. And once you go down the hill, that's where you end up with your product. So you have a start where you want something to happen, and you build up your activation energy until you have enough, and then you go down the hill. And that going down the hill, the enzyme has had the substrate put into its active site, and now something is going to happen, and you'll end up with a product. So that's one way you can visualize it to help make connections. I've also included on my notes that the substrate is specific. It's a specific molecule that an enzyme will act on in a chemical reaction. So the substrate is just as necessary and just as specific as whatever enzyme you happen to be working with. So here's just a close-up of the notes again. Your notes do not need to look like this, but the content should be similar. Just because it's very important that you are intentionally taking notes. If you are not taking good notes, you can't use these again. So I've extended my notes even because I know about chemical reactions. So I'm going to include things about chemical reactions. Well, without enzymes, the reactants would need much, much, much more energy. They would need more things included. So extending my roller coaster metaphor, if we didn't have enzymes, that active energy that we were building towards to get to the top of the roller coaster, we would need to climb much higher in order for that reaction to occur. And that would mean if we're talking about humans would need to consume more energy. We would need to spend most of our time just eating and drinking to get enough calories to change things. So it has been nearly 15 minutes. My timer's about to go off. Let's start that banana phenomena echo now. So we've taken some notes and now we're coming back, we're revisiting. So go back to your initial page about that phenomena study, our banana slice. So after 15 minutes, this is what my banana slice and the peel section looked like. If you notice, we have changes. This is browning. It looks different. It might feel different. It might smell different. So what do you notice and what do you wonder? So go back to that page that you started your phenomena study on, and you're going to do another I notice and I wonder now that it's been 15 minutes. So here's what my page looks like. I still have my initial I notice and I wonder, and now it's been 15 minutes and I've noted the time because I don't have to memorize everything. I can write down my notes. And I notice the banana smells sweeter. It smells more banana-y and it feels a little slimy. It feels different. Before, it had a dry feel on the sides. It no longer has that. And it just felt wet at the cut side, that top side. But now it feels slimy. I don't want to touch it. My I wonder has changed to I wonder how these changes take place in a dead organism. Stuff is changing in this banana slice, but this banana slice is no longer attached to the banana plant itself. So how is this happening? How are enzymes still doing this process? So again, your notes do not need to look like mine, but you should have two separate I notices and I wonders. All right. I know we've used a couple different metaphors to talk about enzymes, and the lock and key is going to be the most common. So I want to make sure we really understand how this functional metaphor can help you in science. So let's talk about it with bananas. So in your banana phenomena study, you saw a change, you noticed a change, and you wrote about it. Maybe you drew a picture, maybe you were very detailed and wrote many things. Maybe you were not so detailed, and that's not great, but it just happened, so it should be fresh in your mind. So using our lock and key metaphor, the lock is going to be the enzyme. So in this case, it's polyphenol oxidase, so PPO. And it means that oxygen is interacting with polyphenol. That's all. Your key is the substrate, and that's the polyphenols, which are natural compounds inside the banana already. Those compounds are inside the banana, but without oxygen, they're not going to do anything. So do you see how the oxidase is going to be changing up what's happening here? So when you cut a banana or when you bruise it, you're changing the cell structure. So the product, what happens when you introduce this key to the lock is going to be the brown pigments. It's melanin-like compounds, so the same thing that gives your skin color. And the keyhole, your active site, is on that PPO. So that's really where oxygen can bind in, is the best way to think about it for right now, to not go crazy bananas here. So at this point, maybe your notes have extended and you're explaining in your notes that lock and key, extending the metaphor. So your lock is the enzyme, and it's the PPO, or the polyphenol oxidase. Your key is the substrate, the polyphenols. Your product, those brown pigments, what changes the look of your banana. The keyhole is going to be the active site on the polyphenol oxidase. So where it can happen, where that substrate, the polyphenols, can introduce or interact with your enzyme, the polyphenol oxidase. So the steps that happen for this lock and key to take place, well, your banana can do this naturally. It can just ripen on the counter, or we can cut the banana and speed up the process by letting more oxygen interact with your cell walls on the banana. 2, oxygen, that O2, has to mix with the substrate, the polyphenols. So that's how the polyphenol turns into, which is a natural substance in your banana, it's how you turn that polyphenol into polyphenol oxidase by introducing oxygen, O2. Your third step is going to be polyphenol oxidase being there. It's the enzyme, it's a protein, and it fits with the polyphenols at the polyphenol active site. The key goes into the lock. And this isn't going to happen just anyway. So you need to have oxygen. That O2 has to be introduced to the polyphenol. So if you don't want your banana slice to brown, you could deprive it of oxygen. You could cover it in lemon juice. You could even cover it with plastic wrap, and you would slow that process down because you're changing the factors that are interacting or impacting your enzyme. Remember when we talked about the pH? So if polyphenols are also in apples, so if you don't like your apple slices to look brown if you cut them in the morning and you eat them at lunch, you can just add some lemon juice, and that lemon juice will change the pH of the polyphenols, and your that enzyme isn't going to be able to interact with the substrate as easily. It's going to change the factors that impact your enzyme. So if you don't change anything, if you don't change the temperature, if you don't change the pH, well, you're going to trigger a chemical reaction, and that's going to turn the polyphenols inside the banana or the apple slices into quinones, and those polymerize. Remember, we talked about polymerization in biomolecules section? You're going to polymerize into brown pigments. It changes how it looks. And so it's unlocking the lock. That's when you're getting those brown pigments occurring. Now, something that I want to make sure that we are revisiting and not ignoring, the enzyme PPO, polyphenol oxidase, it doesn't get used up. And so it can be reused. It's why the banana will keep ripening. The longer you let this sit, the more brown pigments you will see. And that's going to change how it looks. So if we did another I notice and I wonder in 15 minutes from now, what would you expect to see? More brown pigments. And that would continue over time. So let's pause and let's preview how this is going to impact some of the other things we're going to learn about in this unit. So enzymes are very important, and they're not just important for humans because we use enzymes all the gosh darn time, but we're also going to have enzymes in photosynthesis. So chloroplasts and other light-dependent reactions are going to use enzymes to help convert the sun energy, the radiant energy, into chemical energy, that ATP energy. So enzymes are necessary there. They're in plants. They're helping speed up photosynthesis so it doesn't take a long time. It's why our radiant energy is such a reusable energy, because we have unlimited amounts. It starts our trophic levels because we have so much sunlight. So enzymes in photosynthesis, they're going to break down the water, and they are the formation of our glucose in our Calvin cycle, which we'll learn more about. Nobody likes learning about it, but it's not a terrible cycle. We also are going to want to make sure that we are aware that we're going to have to balance some equations because we will be seeing conservation of mass. You can't destroy or create anything, but you can recycle it in some unique and interesting ways. And we'll see the role of enzymes in both phases. So photosynthesis and cellular respiration are always talked about at the same time because one tends to be part of the other. And you'll see that in another slide, how closely related they are. But here you get an idea of it. We have one side of a chemical equation for photosynthesis, and the other side is perfectly balanced with it. So we're using the stuff that goes in the products, and what's on the product side is on the reactant side. It just looks different, but we're reusing the same atoms. We're just organizing them a little differently. So not just photosynthesis, but also cellular respiration. Said they always are talked about at the same time. Cellular respiration is going to take place in the mitochondria, whereas photosynthesis, that takes place in our chloroplasts in plants. In our mitochondria, enzymes help with glycolysis, the Krebs cycle, and that electron transport chain that you should have heard about in 6th grade. Conversion of glucose, sugar, to energy, ATP. So we'll get into more vocab with the cellular respiration side, and it's not hard. You've seen most of these: mitochondria, the powerhouse of the cell, ATP energy, oxygen, carbon dioxide. We just want to make sure we can identify these very easily because that will make your life a lot easier, especially on that unit test coming up where you will see different equations that are they balanced or are they not balanced. So the products of one are going to be the reactants of the other. That's why photosynthesis and cellular respiration look so gosh darn similar. What's on one side tends to be what you see on the other side. So photosynthesis, that makes sugars, glucose. Respiration, cellular respiration, it breaks down glucose for energy. So we break down sugar to make our ATP, and that happens in our mitochondria. Whereas making the sugars in our plants for photosynthesis, that's happening in the chloroplasts. Enzymes drive both of these processes. Otherwise, for cellular respiration in humans and animals, we would have to eat an enormous amount to get that energy. Enzymes mean that we can be far more efficient. Same thing for our plant friends. Their chloroplasts do not need to eat as much sunshine because enzymes are going to make photosynthesis a more efficient process. So now you have a CER activity prompt, and I think you'll like it. We're going to talk about the enzyme lactase and how it breaks down the substrate lactose. So if you've heard those words, it's because they're usually associated with milk. So you're going to have a medical study and you'll hear a little patient backstory, and then a doctor's going to make a diagnosis. Your job is to figure out what evidence was used to support the claim and the reasoning that are already provided. You'll submit this through Canva. So we've done that before, and if you forgot, I'll remind you in class. So you can use this link or you can use your Canvas link to find this CER. But this is going to be something that is checked, so you need to do it. Do not forget to do it because it's so gosh darn easy, and it will help you review that lock and key and how enzymes are present in a lot of different ways. Also, you have a reflection prompt. We've done these in other instances during class, but for this I want it to be enzyme specific, and it also is going to be a little just check-in. I want to see how you're feeling about this material and also maybe a preview of a potential unit test question. So we will talk about this, and I can't wait to see some of your responses, especially on that CER. I'm excited to see how you guys interpret the information provided. All right, bye.